Morning, everyone. I'm Kai Yuan, a postdoc from Dr. Hailian Huang and Dr. Jiange's lab. Today, I'd like to talk about our recent developed cross-population fund mapping method, and entitled with its fund mapping across diverse ancestries drive the discovery of putative causal variants underlying human complex traits and diseases. Before our talk today, I'd like to give some brief background. First, how can we locate some causal variants? During the last almost 2 decades, the GWAS have achieved a great success and identified more than 100,000 of significant variants. But fewer of them have been resolved to maybe functional or specific function. So majority questions remain for the GWAS disease or trait association. So there are 3 kind of questions. First, what variants are involved? And what kind of biological function do these variants interrupt? And what is the disease causing mechanism underlying this variant? So today, we will talk about the first question. What kind of variants are involved? And to solve this kind of problem with a series method called fund mappings were developed. So in this slide, I'd like to give people some intuitive understanding about what is fund mapping. In this figure, there are 3 panels. The first panel is a true effect size, which is unobserved for people. So in this region, we do find 2 causal variant signals, and they are in 2 different colors, red and blue. And the second panel, B panel, is the GWAS Manhattan plot. And the y-axis is a log10 p-value, and the x-axis is the different positions on the genome. We do see a lot of significant signals in this region due to the LD something. And the panel C is what we do for fund mapping. After the fund mapping, we identified some kind of causal variants or candidate causal variants from this GWAS signal together with the linkage disequilibrium. And we found for the second signal, we resolve this signal to successfully resolve this signal to a single variant resolution. But for the first one, the red one, due to the linkage disequilibrium and the limited sample size, we don't have enough powers to have that higher resolution, but it's still much better than the GWAS signal. So this shows what is a fund mapping, and our purpose is to find some candidate causal variant for the downstream functional analysis. Some overview of the current or existing method for the fund mapping. So generally, they can be divided into 2 parts by the number of populations or number of data they use. For the left one is single population fund mapping. That is easy to understand. They use the data from single populations. And some methods, a lot of methods have been proposed, like FundMap or suSiEx or something. The rest of 3 are the methods widely available or widely used for the cross-population fund mapping. The first one is quite intuitive, quite straightforward, the meta-analysis based method. People do meta-analysis before they do fund mapping and makes the data looks like a single population. And for the LD, it's hard to get some average or something. People use maybe some sample size weighted to average the LD or average the correlations among different variants. And second one is maybe some single population combining method. This method is also straightforward. They do a single population fund mapping separately for different populations, and then they using clustering method to combining or merging the different critical sites together. Last one is maybe some model-based, some Bayesian statistic method. There are 2 existing Bayesian cross-population statistic method for Pinter and MSKVR. So here comes to our model. Let's start it with the most simple model, our basic, the most simplest one. Single effective regression model, SER model. So firstly, there are several symbols for Y. We use this to stand for the phenotype, and the X is the genotype. X is a matrix with the n different sample and the p different p and the p variant, and the B stands for the effect size. Effect size. This is actually this is a vector, and this vector is determined by the B and the gamma. Gamma is also a vector to determine underlying true causal. Gamma follows a multinomial distributions, and there is only one exact element equals to 1. All the rest are equals to 0. 1 stands for this variant is causal, and 0 stands for non-causal. So in the B vector, there is only one element is non-zero, and this stands for the non-causal. Sorry, this stands for the causal. So that's the means for a single effect. And E stands for the noise. And we're using some normal distribution to model this. So in summary, for the SER model, there's only a single effect, means the vector B has exactly one non-zero element, and this equals to the B, small b. Small b is also follow the normal distribution. And the sum of a single effect model, actually this model was proposed by Gao Wang proposed by Gao Wang, and this method is called SuSiEx. In their model, the key idea is also simple. They introduce L different effect vector. So the idea is that there could be maybe multiple causal variants in each locus or in each regions. How do they model this? They actually model that there are L different vectors. Each vector represents one single effect causal variant, and they sum all the different B vectors together to get a final B. So that's the means for sum.
Sorry, a quick question. Isn't there like a window size which you select and then look at the variance for this, and how do you select those window sizes?
That's a good question. Actually, mapping did after the GWAS. For GWAS, we identify some loci with significant signal. Yes, we believe this region do have maybe some causal or something, because we do observe some significant signals, but we don't know which variant should be responsible for the causal. So for mapping, we already did this. So here comes to our model, suSiEx model. In previous SuSiEx model, they cannot handle the data or results for different populations. But in our model, we extended this in by adding new dimensions. So here we introduce the S indicate the different populations, and we assume all the causal variants across different populations are shared, which means the gamma L stands for different signals. Gamma L doesn't depending on the S, so the causal variants should be shared across different populations. But here we allow the effect size.
So is the assumption that the variant will have the similar impact on different population, or because it's not necessary, like different population, the variant may impact the same different population differently? So what's the assumption here? Good
question. Actually, here we assume the causal are shared, but we allow the effect data. The B could be different across different populations. Yeah. Also, this could be the B different could be some population could be 0 or even closer to 0, which looks like it's population specific. Yeah, but we will show this later. These assumptions will not affect our model too much. We'll show this later. Good question. Thank you. And then here comes to our simulations. We use simulations to so we use simulations to test our software. For our simulation settings, we randomly generated 101 megabits regions, and each region there the manual frequency we take the so for our simulation settings, we randomly select 101 megabits regions from the genome, and we keep the variance with the manual frequency greater than 0.5%. And for each region, we simulate one causal variant, and the cross-population genetic correlation is 0.7. Probability we set is 0.1%, and for each simulation settings, we replicate 5 times. And in this these figures, we show that first we show the power. Here we define the power is the number of causal variants identified or discovered by our method, or covered by the critical site. Here in this figure, the lower panel shows the sample size we used. European, African, and East Asia. The larger dots stand for the larger sample size with 100,000, and the smaller dot stands for the smaller sample size data. And the first one is the same. The ground truth was same with different colors stand for different variant type. If this variant is only maybe greater with the manual frequency greater than 0.5 in European, we say this is a European variant, or African variant, or East Asia variant. For all the rest, we share this. We said this is a common. We use this to label different colors. And the first column is the ground truth we simulated the variant. And here we use the 50,000 European data as our baseline. After we adding 50,000 more data from both European, African, from European, African, and East Asia, we found that the power increased. But by adding data from the different populations, the power increased more. And the best performance result is the 150 data. From 150 sample size results, we combine all the data together. That's outperformed by all the others.
Could you perhaps please say a bit more about the cross-population value? I'm just trying to gain some intuition rather about where the 0.7 would come from.
We can just so this is a cross-population RG genetic correlation is underlying effect size the genetic correlation. We're using the multi-normal distributions, and this is the genetic correlations of the different populations of beta. So all the underlying effect size.
I think that really helps. So for example, if the effect size were identical across populations, that would be 1. Exactly. And if it is not at all correlated.
Yeah.
Got it. Thank you.
Something along the same line, I'm going to ask the same question in a different manner. We know the European population is well studied, so there will be much more samples than African or East Asian. How are you not normalizing, but maybe normalizing the effects, understanding the effect of the variant, where the sample size is completely different? So the effect side will be also different. Yeah, so I think I'm asking the same question in a very different manner.
Yeah, so we will consider the sample size in our result. Yes, I understand your question. Maybe they're really unbalanced results. In our later empirical data analysis, we use the UK Biobank. This is the largest sample, largest data set. And by adding data from other population, we do find some improvements. Even this is a very, very tiny, or not tiny, even it's smaller than UK Biobank. Yeah.
Is the assumption correct that you are looking for more, because myelin frequency of greater than 0.5, you're looking for non-rare variant, you're looking for more common variants? Yeah, exactly. So our purpose is to find maybe some, so first we will not aim to maybe find some rare causal, because it's harder to capture this both in the LD calculations, maybe for the rare variants is hard or difficult. And also the regression for the rare variants is also difficult. So our purpose is to aim to some common causal variants. So here comes to the coverage thing, or we also define this as a calibration. The proportions of credible sites that include at least one true causal variant. You can also think this is some kind of accuracy. Yeah. Here, we use the alpha levels of credible sites. That means the coverage, we should expect the coverage for each credible site should be greater than alpha. So we set this, we set the alpha as 0.95. So our coverage also shows that the accuracy is quite high for our method. And the resolution is also quite an important thing. So we define the resolution as the size of a credible site and the number of causal and the number of variants in the credible site. Here the size of a credible site, you can say as we add more samples or
more sample from different, for diverse populations, the size of critical sets also decreases, which means the resolutions will increase by adding more data. And also the number of causal variants with extremely higher PIP, which means this variance we are highly confident. We count the numbers of a PIP with greater than 0.95. And the numbers, confident variance also increased by adding more data. We also do some more exploring with effects of different parameters of simulations. As you're being asked for the genetic correlations, we set it for three different values for 0.4 and 0.7. 0.7 is our standard setting, and also exactly genetic effects for 1. And the power, coverage, and the resolution are not affected by our genetic correlation across populations, since we allow the effect size could be different across populations. So this will not affect our model. And heritability we set it for different values for this. This intuitively, the higher heritability will have a larger power and a higher resolution. And the number of causal, we set it for 1, 2, and even to 5. And in our method, we're able to find more numbers of causal as the NCSL increased, and all the I think all the critical sites are well calibrated with the 95% coverage.
Hello. If you have a causal variant with different allele frequencies in the two populations, then if like the per allele effect size is the same, then the heritabilities will be different just from that variant. So is this like, what is this heritability number?
So actually, we didn't take that stringent heritability calculation. We just maybe using a very, very simple one to generate some data. Actually, I do agree that allele frequency will affect the real heritability thing. Yeah. So here we want to just emphasize with maybe the trend for the higher heritability, a better performance of a method. Yeah. We do agree that the heritability is not that accurate heritability. Yeah. Thank you for your comment. And then we also want to test our model assumes that the causal variants are shared across different populations. What if it is not shared? It's population specific. In this model, we test we perform first we perform single population from mapping analysis for 50,000 European and 50,000 African. This is our baseline. Then we generated some non-data. Non-data means they carried 0 causal. That's a noise. We added the 0. So here the gray dot stands for the non-data. After we adding some non to the result to our software, I think the power reduced very, very little, and we can maybe just say it's not affected greatly by the non-data. And also the coverage is quite good. All maybe close or above the 95%. Then also we want to dig into a little bit more. So if we know this is a population specific thing, if this is a population specific causal, can we maybe identify which population they manifest the causal? Then we propose the post hoc probability to determine whether this critical site is shared or specific to some specific populations. Here, the calculation is simple. For each critical site, for 2 populations, there could be 4 different kinds of causal configurations. Here we define the configurations means like this variant is so for first one is a POP1 specific, and the second is a POP2 specific, and the 3 is a shared, and the last one is a non-causal for both populations. Then we calculated the post hoc probability of each critical site being causal under each configuration. So we level with different P1, P2, P3, P4. And for POP1, we only add P1 plus P3. POP2 is P2 plus P3. And we use simulation to test the performance of this kind of post hoc probability. In this scenario, we use a 50,000 European data plus 50,000 African non-data, which means this causal variant should be European specific. And the left panel shows the probability in European. It should be higher, right? And in African should be lower. We use the 0.8 as our threshold. If this is above this 0.8, we say this is the this variant manifests causal in this population. If it's not, we show this is not causal. And the simulation results show that for the European specific, there are 100% variants. We identify them as causal in European specific. And the African, there are only, I think, two causal variant, two results that were falsely reported as causal in Africa. Similar for African specific scenarios, it's quite higher, more than 99%. And only one variant manifests falsely causal in European for the African specific thing. Also, we want to compare our method with maybe some commonly used or more intuitively method meta-based SuSiEx or meta-based method. Here we for the right one, we use the meta-based SuSiEx as our comparison target. So we do the fixed effect meta, combine the data across different populations to get the GWAS summary statistics. And for the LD, we're using maybe some sample size weighted LD. We combine the LD for different populations. And we perform the single population file mapping SuSiEx to inform the results. Here's the power comparisons between SuSiEx and the meta-based SuSiEx. Here, the bottom also shows the sample size, and here different color for different type of variant, and we can clearly see that the SuSiEx outperforms than the meta-based file mapping for the power. And also, this is quite interesting for the coverage. We found that there is a greatly coverage loss for the meta-based file mapping if we have a larger data set, large sample size. For SuSiEx, all the coverage is quite higher, about maybe above the 0.95, but for 400,000 data for meta-based analysis, the coverage only 50. So brief summary here. So SuSiEx is so we can have a larger sample size, and if we have a more diverse populations, we will have higher power and higher resolution. And the coverage of SuSiEx is well calibrated, and the genetic correlation doesn't affect SuSiEx. And SuSiEx also can identify population-specific causal variants. And meta-analysis file mapping will lead to power loss and bad calibration. And then we compared our method with the existing method for cross-population file mapping, Pinterest and MSKVR. So here, firstly, we defined 3 different running status. So for the green color, we use this for the successful finish in 24 hours with 1 CPU core, and returning the sum of PIP is between 5 and 0.1. We do this because some software returns some unreasonable results with every variant or every marker with 0 PIP or 1 PIP. That's totally unreasonable. And unfinished means the runtime is longer than 24 hours. This happens in the MSKVR. For the running status, SuSiEx performs good. For we successfully finished in 24 hours, usually just maybe several minutes or several seconds. And the source for the unreasonable thing is mostly come from the Pinter, especially for the 4 variant size. And for the MSKVR, the runtime takes extremely long. They can only maybe handle the variants with at most 150. Yeah. Then we use this figure to compare the performance. The x-axis is the number of identified the causal variants with the PIP greater than 0.5. Greater than 0.5 means we can find a dominant variant in each critical site. And the coverage also same definitions or something like the accuracy. the dashed line represents the 95% calibration. And we can see the pincher and the suSIEx is good, but for the MS-Caviar, there's some problem with actually the resolution. MS-Caviar do identify a lot of causal, but the critical site is extremely large. Since we only use 150 variants for each region, they often return maybe some large critical site with greater than 50 variants. So this is too large, too big. And by comparing the pinchers and the suSIEx, you can see the power or the axis is axis comparison for the suSIEx and the pincher. suSIEx is a little bit better than pincher, but there is also a problem for suSIEx. When we use only 150 top variants, the coverage of suSIEx dropped below 0.95. We believe this is due to maybe we removed too much variants, and I think it's totally okay if we just give them 4 variants in this. Then we applied our method to some real data. We do this for the UK Biobank and the Taiwan Biobank. For UK Biobank, we take the European data and the African data. And also the Taiwan Biobank, we take data from, this is the Biobank for the East Asia. We applied our method to 25 overlapping quantitative traits. And there are more than 14,000 candidate loci, which manifest the significant genome-wide significance in both single population or cross-population meta. So in this figure, so the left panel shows the maximum PIP of each critical site between the suSIEx and the European single population from mapping. And we can see the red dot stands for the average value and the box stands for the median. We can see both for the median and the average, suSIEx is better than single population suSIEx with European. Here we compared this with the single population European because the European data is the largest data. And if we see the right panel, you can see that there are only limited numbers of credible sites with high confidence identified by African and East Asia cohort. So we only compare the cross-population suSIEx with the European, cross-population suSIEx with the European suSIEx. And the right panel shows the number of credible sites with our highly confident with PIP greater than 0.95. Also, suSIEx is the best. Then we align our suSIEx for the credible site with a single population identified credible site. The x-axis shows the maximum PIP for suSIEx, and the y-axis is the maximum PIP for 3 single population from mapping. And we observed most of the credible sites were improved by suSIEx. And we do find some credible sites with great PIP loss. But further dig into this variant or this causal variant, we found this candidate region. We found this region might be with some genotyping errors. Yes, there are a lot of indel or something there. We're using maybe imputation data. If there are regions are in range of indel or something, the imputation quality might be some problem. And we further dig into this kind of quality issues. We found that the y-axis is the proportions of credible sites with maybe quality issue. Okay, so the x-axis is the maximum PIP loss from the single population to the cross population. So the left one means the PIP loss is minor, and the right one is have a greater loss. So as the PIP loss increases, the proportions of credible sites with quality issues also increase. Then we compared as the PIP increased, there are also some enrichment of the high PIP variants with a high or moderate functional impact. Different color levels for different kind of categories variants. Red stands for high, and yellow stand for moderate, and moderate includes the missense variant, and the low stand for some maybe minor and modifier. Brief summary here. So cross-correlation file mapping in the Bell bank shows that the power will be increased. We have the ability to find some novel critical sites which cannot be detected by the single population file mapping, and the resolution also higher. The higher maximum TIP and also smaller critical sites. And we can also find some population-specific causal. We will show some examples later. So here is our highlight examples. In this region, this is a phenotype for the total bribery. And here, this figure shows the GWAS for 3 populations. The first panel is the European GWAS, African GWAS, and East Asia GWAS. And we can see for the European, there is a quite insignificant signals here. And this is also a message variant. And for the African, there is a bunch of variants achieved the genome-wide significance. And in East Asia, there is one single variant in the intron. Then we do the file mapping. In European single population file mapping, we find nothing. And in African, we do find this bunch of significant variants, but the resolution is not that high. In East Asia, we find these signals, but it's a 2 variant signal, but dominated by 1 variant. For the cross population file mapping, we find 3. The first one, the last one, this variant cannot be identified in either of the 3 populations. We say this is an example of a higher power. And the other 2 is the population specific thing. One is the green one is the East Asia specific, and the blue one is African specific. And also we can see the PIP also with a minor increase from the single population to the cross population. That's a small. And also these 2 are the examples of the population specific.
A few slides back, you showed the quality, the sensitivity, and you mentioned that out of 123, 2 variants still pass your threshold in European. Remember, it was just the European and an African you test. So in European, it should be 100%, but you looked at in African, you still found 2 variants. So you had some false positive shown in your case. So when you're showing this, how can we confirm? Yes, if you go back here. So yes, so given that there are cases where you will find some false positive, how can you confirm what you are gaining over there is a true signal?
Yeah. So first to clarify this is we see this a post-hoc probability. This is something after we identified this as a causal, then we calculated the post-hoc probability for whether this variant is specific to each population. There has some false positive. And what you mentioned for the false positive may be related to this kind of coverage. Yes, well, yes, we do have some false positive thing, but yeah, we cannot 100% this is a causal, but we can say this is some candidate causal. Yeah. So if we do.
So taking this information and the issue with genotype, where the genotype error, you did find genotype errors.
Yeah.
What sort of QC steps we should take to reduce these type of? Do you have any recommendations?
Yeah, good question. So I think to do file map, it's always good to maybe take some stringent QC to remove a variant, but it's also some balancing thing. I think it's hard to find some golden standard. But for file mapping, I think we suggested to remove we take some stringent QC to remove some variants with maybe some clearly error or something. Yeah, but always cannot maybe remove every error or genotyping error from the data. Yeah. Also, continue to our example here. we also calculated the post-hoc probability to determine whether this variant is causal or manifest causal in each population. For first variant, it calculates the post-hoc probability for African is equals to 1. That means this variant is manifest causal in only African. This one, the left one. And in the middle one, this manifest causal in East Asia, but not in European and African. For the last one, that's quite interesting. This is variant is shared for European and East Asia, but in African, we didn't have enough we don't have enough confidence to say or support to say this variant is manifest causal in African. The p-value also supported our results that the p-value for a negative log 10 p-value for African is only 1.62. But maybe there is some weak signals here, maybe. But yeah. But this variant didn't meet the threshold for African specific. And we also applied our method to the schizophrenia, so the European and East Asia data. We compared our results with previous published Nature paper, the PGC3, they use the fun map and the meta-based fun mapping. They first they do the meta and then they perform the single population fun mapping with the fun map. Compared with a number of credible sites with different confidence. So the left one is a maximum PIP greater than 0.95, and the right one is the maximum PIP greater than 0.5. Different color scheme for different software. We can say our method is better than the fun map, especially for the variants with the maximum PIP greater than 0.5. And brief sum here. So SuSiEx could take the input from the summary statistics plus LD, and also we can the data could be from the different populations of different ancestries. And the SuSiEx is better than current or existing Bayesian cross-correlation fun mapping method, Painter and MSKV, by user-friendly and higher power and higher resolution, and also higher computational efficiency. And the SuSiEx is also facilitated to discover the causal variants and improve the fun mapping resolution in both biobank analysis and PGC analysis. Here, the last slide, I'd like to thank my advisor, Dr. Halian Huang and Dr. Tian Ge, to design this project, and also thanks Ryan to do extraordinary work of SuSiEx, and thank Jiayan Chen, and thank Max Chen Yu and Minrui Yu. And also thanks to our amazing collaborator from Penn UK Biobank and Taiwan Biobank. Here, to scan the QR code to get our latest updated software. And we have just updated our method from the Python to C++ this week. And this software is much faster, and the memory consumption is highly reduced. Yes. So if you're interested, and you can also see you can not see the link, but you can search our preprint on the Meta archive. So thank you for your attention.